Welcome back, friends. In this video tutorial we'll be talking about the Streptococcus pneumoniae, and it's not about Streptococcus pneumoniae in whole. We'll be just looking at the pathogenicity of Streptococcus pneumoniae, how they brought about the pneumonia, that means the virulence factors of Streptococcus pneumoniae to cause an infection properly in human body, and how our body responds against that different approach of the bacteria, and finally the treatment of Streptococcus pneumoniae infections, that is treatment of pneumonia in very little extent. Majorly we'll be focusing on the different virulence factors and the approach of the Streptococcus pneumoniae to set the infection in human body. So let's begin. So first thing about the signs and symptoms and type of diseases they cause. Now normally the Streptococcus pneumoniae are the normal inhabitant of upper respiratory tract of human body. They normally resides there just like normal flora, but they are opportunistic in nature. They are opportunistic pathogen in nature. So sometimes they can cause infection, they can cause diseases when situations are turning to their favor in many aspects, right? And the pneumonia they cause usually of lobar type of pneumonia. There are many variety of pneumonia, but the streptococcal pneumonia is a kind of lobar type of pneumonia, and also they can cause paranasal sinusitis and they can also cause meningitis, which is usually very secondary, and it usually reach at the end point of the infection, but it's very rare when they crosses that blood brain barrier and can reach our meninges, when they can cause the infection and inflammation of the meninges. It's very rare case, but usually they cause pneumonia and sinusitis. Now this streptococcus pneumoniae currently is the leading cause of invasive bacterial disease in children as well as in elder, in both the way. Let's talk about the infection overview and the pathway of their pathogenicity in detail. Now we can divide their pathway of pathogenicity into three different sections. One section is dealing with the pathogenicity occurs at the very earlier stage during the mucosal surface or the mucosal epithelial surface. Second stage of the interaction is in the bloodstream, and the third phase of the interaction is in the cerebrospinal fluid if it reaches that meninges. So let's begin with this first part, which is the mucosal epithelial damage. Now in that case what we know, they are having different factors. Now those factors are helping this bacteria to produce to actually evade the normal immunity of our body, to degrade antibodies, to break down any other cellular toxic compounds and everything. So usually what we can see here, NAN-A is a type of protein secreted by Streptococcus pneumoniae, which is a N-acetyl N-acetyl acid from mucin, right? So they start to secrete those things, and this NAN-A is and also along with another protein which is called IgA protease. Now as we know, this is a protease, that means it is going to cleave proteins, and in this IgA1 protease is going to cleave IgA1 or immunoglobulin A1. So they will cleave the immunoglobulin that is present in mucosal epithelial surface, and also NAN-A is kind of degrading the mucin barrier there or the mucus layer there. Now the second round is in the bloodstream, and in this case they also produce some capsule outside their cell membrane. They are having a cell wall, and outside their cell wall they are having a coat, glycocalyx kind of layer. It is called capsule. This capsule is very important to help them not to be opsonized, not to be phagocytized, because for the phagocytosis they require complement system protein C3b, but this capsule prevents the interaction between C3b and phagocytic receptors and blocking the opsonization process. On the other hand, they can also produce another factors like PSPA, which prevents the activation of C3b, right? And also it can produce PSPC, which is again binding factor of H, and it also prevents it also favors the degradation of C3b. So you can see in all these aspects, producing capsules, PSPA molecule, PSPC, PSPB molecules, what they are doing, they are actually minimizing the interaction of C3b with the phagocytic receptors or phagocytic cells, and also sometimes they're degrading those C3b. They are also producing hyaluronate lyase. This hyaluronate lyase is going to break down the hyaluron or hyaluronic acid that is present in the tissue lining of the subepithelial extracellular matrix. So the subepithelial extracellular matrix is kind of broken down due to the secretion of this kind of hyaluronate lyase kind of molecule. This Pneumoniae bacteria, they also produce a very important mediator which is called pneumolysin. Now this pneumolysin can bind to the Fc region of IgG antibody, and what it does, it is kind of lyse that IgG antibody, it is kind of cleave those IgG antibodies. Okay, so these are all the sequential stages that are occurred in bloodstream all the time, because this is the phase where the bacteria start to evade our system. It needs to elude our immune system and spread very quickly. This is the rapid spreading phase of that bacteria, and it is producing several different proteases, several different enzymes to block the different activity against this bacteria by or imparted by our body. And not only that, but they can also shift from a phase variation. Now that phase variation will certainly change the type of outer cell proteins they are having. As a result of that, the ability of binding with different receptor for that bacteria kinds of change. Okay, for example, pneumococcus can bind to carbohydrates like galnac. It can bind with CBPA, it can bind with kind of PAF receptor using another protein called CBPA. But the variation between the binding of with different type of receptors and receptor molecules onto the host cell layer surface, it varies depend upon the need of that bacteria. It can actually vary those phases of its cell membrane proteins or cell membrane receptor or binding proteins. Now if we enter into the third phase, when the bacteria kind of crosses the blood-brain barrier, prior to reach the blood-brain barrier, it must be adhered to the blood-brain barrier layer or epithelial cells. Now for that epithelial cells to be attached, that's why they need to bind with carbohydrate like galnac. They need to produce CBPA proteins so that they can bind and pass through PAF receptors and finally come into the cerebrospinal fluid. Now once they are successfully made that happen, they are successfully entered inside the cerebrospinal fluid. In that condition, they start to spread again, because in this condition the local immunodeficiency allows uncontrolled bacterial replication, and bacteria start to divide and grow and thrive in it many numbers. Now this is the situation when the bacteria is kind of taking the immune system against our body, and also none of the immune system can reach there because it is the cerebrospinal fluid layer. And then they start to produce further type of autolysin like light A, light P. So the autolysins, the streptolysins start to produce. They are functioning like exotoxin. They are degrading the tissue, they are damaging the tissue there, and also they can kind of form inflammation there, and they start to evade the antibodies also in this particular case. Okay, and also the breakdown product of bacterial cell, the subcellular, subcapsular bacterial products can also create damage in this particular section. So this is how three different way this infection can reach the cerebrospinal fluid and finally to the brain. Now till now we have talked about how this Streptococcus pneumoniae start to produce an infection. Now we must know that body's defenses against that Streptococcus pneumoniae, to control it, to prevent it. Body is also creating several different rounds to control this Streptococcus pneumoniae. The first round is to block the Streptococcus pneumoniae in intracellular killing. When the pneumonia at the very beginning adheres to the mucosal layer, they try to destroy them by engulfing them, endocytosing them, and degrading them by the lysosome fusion. That's the normal approach. But if this never happens, if the bacteria enters inside the cell and it reaches kind of extracellular matrix layer, at that point they start to produce some antimicrobial agents always. Those antimicrobial peptides, antimicrobial proteins and everything, they start to produce them to degrade those cells to control their growth and division. But if again it fails, then the third layer of response is also there, which is the inflammatory response. When the bacteria reaches the epithelial cell through the basal layer, when it reaches at the inside the bloodstream, they need to use normal immune system cells to prevent it, and during this point inflammatory response set place. And for that part to occur, they will call upon many important chemical mediators and neutrophils, basophils and all these molecules to finally regulate the growth, and it causes the inflammation there. But still, if this inflammation fail to check the growth and development of Streptococcus pneumoniae, then finally the innate immunity plays an important role. The complement system will play the important role. The C3b protein of complement, the immunoglobulin A of our humoral response will come and bind, and the phagocytes will take upon their job. But remind you, we have already talked about the Streptococcus pneumoniae can produce several ways to bypass this innate immunity responses, because they are producing some proteases that will cleave IgA. They can produce some proteases, they can cleave the Fc portion of IgG. They can produce some factors that can degrade C3b or that can inactivate C3b, so that they can evade this whole body response against them, and they can successfully cause the disease. Now let's talk about the pathogenicity of factors, or pathogenicity factors that are responsible for creating the successful infection for Streptococcus pneumoniae. The first thing I want to talk about is capsule, because the capsule layer is very important to block the phagocytosis process by the phagocytic cells of our immune system. And the capsule is made up with mainly polysaccharide. It interferes with the phagocytosis by preventing the C3b opsonization of bacterial cell, because it blocks the interaction between C3b and phagocytic receptors. As a result, phagocytosis kinds of halt there. And there are 90 different type of capsules are isolated and identified on the basis of antigenic serotype of that organism. That's a fact. Now the second thing which is helping them is again extracellular part, and that is pili. Now pili is helping them for the colonization in the upper respiratory tract, and that is very important. The increase in the formation of large amount of TNF by the immune system during the invasive infection, and this pili will help them to finally attach to this upper respiratory tract, which is very important for them to stay there for the longer run in throughout our body. Third thing, cell wall, which is composed of peptidoglycan, and also they are having teichoic acid, which is attached to the approximately every third N-acetyl muramic acid residue. They can also have lipoteichoic acid attached to the cell membrane by a lipid moiety. Now both the teichoic acid and lipoteichoic acid contain phosphorylcholine. Now this is very essential to have this phosphorylcholine, because the choline especially adheres to choline binding receptors. Now they are having this phosphorylcholine in teichoic acid, lipoteichoic acid, and this choline can easily bind with any choline receptors of your body cell. And in human body we find there is virtually not a single cell not having a choline. So every cell kind of having this choline receptors. So using the teichoic acid presented choline, this bacteria can grow and attach to, adhere to choline receptors present in human body cell. As a result, they can adhere there, and then it will be easier for them to engulf inside, so that they can cause the infection. Then the surface proteins play a very vital part about this streptococcal pneumonia infections, because this surface proteins, some of them kind of proteases, some of them kind of C3b inactivitants, some of them can degrade C3b, some of them can act as kind of lytic agents, kind of proteases. For example, there are almost about 5 different types of surface proteins identified, and that includes 5 penicillin binding protein, and that's very important. And they are also having 2 neuraminases which can break down neuraminic acid, and also they are having IgA IgA proteases, which can break down immunoglobulin A molecule. So you can see they are proteases, they are lipases, they are always present, they try to degrade the protein structure, they try to disrupt the structure and all these things. Now normally the neuraminidase and IgA protease that is present there, these exoenzymes like neuraminidase or IgA protease, they are synthesized and then they are kind of embedded onto the cell membrane of the bacteria, and they are having very vital role in the virulence. Normally this neuraminidase cleaves off the sialic acid residues onto the host cell receptors to convert it to a preferential binding for the pili of the pathogen. Because if this sialic acid residue is present onto the surface of the host cell, it is difficult normally for this bacterial pilus to bind with it and to adhere with it. To remove this difficulty, they kind of cleaves this salicylic acid out, and a unique group of choline binding proteins, so CBPs. This short terms are very much used in this case, CBP, PSP and all this. So CBP means choline binding protein. They are also having them. So they are having kind of 12 choline binding proteins, and they are non-covalently bound to the choline moiety of the cell wall through the common C-terminal domain. And normally the CBP family includes such important determinants of the virulence like PSPA, which is an example of protective antigen, and light A, B, C, which are autolysins, and CBPA, which is again an adhesine. So all of them, this PSPA, light ABC and CBPA, they are among kind of choline binding protein family proteins, and among them this PSPA acts as a protective antigen to help them to be activated against different immune system cells. Light ABC helps them to degrade and lyse several region or sections of our tissue to spread the infection, and CBPA is helping them to kind of adhere to the surface of the host tissue prior to the engulfment of that bacteria inside. Now the protective antigen PSPA, it is known to inhibit the complement mediated opsonization process. So in all the way what we are seeing, there are many important ways to evade our immune system, because normally pneumonia is very controllable disease. This is very common, treatment is always available, but nowadays this pneumonia kind of getting very dangerous because they are modifying themselves and they are finding new ways to evade our host immune system. Autolysin light A is responsible for pneumococcal lysis in stationary phase in the presence of antibiotics and the response to host defensins. Now in this case, this autolysin, they usually secreted in stationary to bind with this in presence of antibiotics, because at that particular point they require to respond against the host defensins, and they do this using a C-terminal domain with 6 choline binding repeats, and normally and an N-terminal domain that provides the amidase activity or amidase activity. Now as pneumococci begin to lyse, they release cell wall components, which is called pneumolysine, and other substances that lead to the inflammation and cytotoxic effect. So you can see here in this diagram, we can easily recognize that different proteases are kind of produced inside, and they are not only produced and placed in cytoplasm, but they are present in this inner membrane space, kind of inner membrane space, because and light A, light B, these are produced inside the cytoplasm, and they are staying there because they require these molecules to go and function when it is required. Now once this process is uptaken by light A during the stationary phase of the cycle, then they need to secrete light B and light C in second phases. That's why the name came ABC, because A acts first, then B, then C, and so on. The B and C will work only after the lysis is completed, and it starts and begin by the light A during the stationary phase of the pneumococcal division. And light B acts as a glucosaminidase, and light C exhibits lysozyme-like activity and then everything will work on there, and that leads to inflammatory response, because they will bring upon the normal immune system cell and the cell mediated immunity of host cell there, and they finally cause cytotoxic effects. Now among them, the CBPA is a major pneumococcal adhesin. We have already talked about that. This is also a part of that choline binding protein family. CBP is a adhesin which helps them to adhere. Normally it is having 8 choline binding repeats, so that's why it can easily go and adhere to any kind of choline receptors that is present onto the surface of the host cell. The adhesin interacts with the pulmonary epithelial surface carbohydrates, and CBP also has been reported to bind secretory immunoglobulin A and complement component C3. It can bind to all of them. Now if we talk about the hemolysin, there are two types of hemolysins that are described. One is the most potent of which is the pneumolysin, which is a kind of 53 kilodalton protein, and it can cause lysis of the host cell. This is the dangerous pneumolysin. It is stored intracellularly and is released upon lysis of the pneumococci. This pneumolysin binds to cholesterol and thus can indiscriminately binds to all cells without restricting to a group of receptors. So as it is having the ability to bind with cholesterol, it can virtually bind with all cells of our human body, and then when it starts to release, it can lyse the whole cell and digest the whole cell. So for the process to occur, they first assemble all those oligomers onto the surface of the membrane, start to form the transmembrane pore or channel, and then cell is finally lysed after the massive water and electrolyte imbalance. And there is another way of having a dangerous effect by this Streptococcus pneumoniae, that is using hydrogen peroxidase, because this pneumococci can also produce this hydrogen peroxidase molecule, and we know these are reactive. This can convert the reactive oxygen species, and this can act as the kind of radical to damage the cell, and starts to damage the host cell, and it can also cause apoptosis in neuronal cells during meningitis. And as a result, for this hydrogen peroxide secretion during the new meningitis, it starts to destroy our neural cells in brain and different sections, and it has bactericidal effect against competing bacteria such as Staphylococcus aureus. So it's very dangerous to secrete this hydrogen peroxidase, because it is not only clearing the path of our human body cell, not only killing the cells, but also it is clearing the path for competing with other bacteria like Staphylococcus aureus. Because if there is infection of Staphylococcus aureus along with Streptococcus pneumoniae, they make ensure that Streptococcus pneumoniae can live through the long run, and it can eliminate the Staphylococcus aureus by the synthesis of this hydrogen peroxidase. Including this pneumolysin and hydrogen peroxidase, both of them using their own method of ability, they can degrade the cell, they can release the cell components and kill the cell components, and it can also induce the production of nitric oxide, which may play a very key role in septic shock syndrome, because this Streptococcus pneumoniae can cause septic shock syndrome. If you look at the genetics of Streptococcus pneumoniae and how they are gaining the different important components to finally evade the immune system and kill the host cells, and that is a unique set of at least 11 proteins preferentially exposed during the competence. That is very important, though it is not completely well studied yet, but it is understood that there are at least 11 to 12 proteins that are present, and they express preferentially during the competence generation according to their need of generation. Early in the competent state, at 17 amino acid peptides start to form, which is called competence stimulating factor or CSP, and it is released from the growing bacteria, and the CSP induces the competence, and when it reaches a particular critical concentration, just like having a kind of system like Coulomb sensing, and that depends on cell density, obviously, right? So as they are producing all of them, they start to get those competence stimulating factors, and other cells start to get this competence factor. And finally, the process underlies the explosion of antibiotic resistance for the past 20 years via the process of horizontal gene transfer. That means via the mode of normal uh, conjugation, transformation, and transduction processes. Now it is found that Streptococcus pneumoniae can also transformed artificially and related to unrelated bacteria. And the upper respiratory tract of the host, horizontal exchange of genetic information can set place between Streptococcus pneumoniae with other bacteria that is present on the upper respiratory tract of human body. So if they are interchanging those materials, they are gaining or releasing some part of their plasmid of having antimicrobial resistance property containing genes. They also develop the antibiotic resistance by mutation and selection, and then they can transfer between themselves, and fast growth rate and large cell densities in an infections setting favor an occasional natural transformation may occur. They can change it, shuffle it, mutation, selection, many things can occur. They can pass through bottleneck during transmission, and everything can occur. And finally, they can set that resistance property. And pneumonia resistance is kind of getting rise nowadays. That's why treating pneumonia is kind of difficult in future days. It is going to be difficult in future days. It is predicted now. But now the treatment is available. The MDR strain is very common, and the range of antibiotics are there: penicillin, cephalosporins, trimethoprim sulfa drugs, cefotaxime, which is the third generation cephalosporin, macrolides, etc. We can use them. Combinational therapy is obviously all the time the best choice, because not one antibiotic can cure it. But if the infection is restricted at the upper respiratory tract level, at the bronchial pneumonia level, lobular pneumonia level, at that point you can use any one two type of antibiotic to treat it. But if it reaches that meninges, if it reaches that uh, meningitis layer, uh, if it passes through that blood-brain barrier, in those conditions you may need to look at much more reliable process to cure it. Vaccine is also available. Anti-pneumonial vaccine is available based on the formulations of various capsular antigens that is present onto the surface of the pneumonia we have talked about. We can derive it. We can usually derive from the highly prevalent strains. We can take it out, but it is not very much efficient. That's why it's not very much heard, but it is available. But normally we don't need to go towards vaccine. We usually treat it. We usually treat it using antibiotics, and it is common. But in future, due to this horizontal gene transfer between other bacteria in the upper respiratory tract, due to random mutation and everything, it is kind of changing, mutating itself, updating itself. So we need to be updated more to get them, hold on to them, and finally destroy them using antibiotic or antimicrobial therapy. So that's kind of it, and I hope it's helpful. Thank you.